This couple is shopping for their wedding cake. So they visit a leading bakery and try out what they have in store. A cupcake. It's delicious, but it's not a given that someone who's good at making cupcakes can also make a wedding cake. For one thing, the scale is much larger, likely made on different gadgets, and some ingredients like yeast don't scale proportionally. So many things can go wrong when making at larger scales. The baker is not interested in making a giant wedding cake just so the customers can try it. Imagine all the wasted effort and money if they don't like it. Instead, she decides to make something in between, like a birthday cake, on a pilot scale. These different scales also play out in pharmaceutical development and manufacturing too. A drug compound is initially made to perform toxicology studies in animals during preclinical trials. The aim is to find out if it shows any adverse effects on crucial organ systems. The volumes of the compound made during the stage are extremely small, produced at what's called lab scale. Just as with the baker, there's no point making large quantities at this stage because you never know if the compound will get regulatory approval. Once approved, the volumes required for market release or commercial scales are vastly larger, usually thousands of times larger than what was made in the lab. You'll need to utilize large-scale manufacturing facilities, play around with different inactive ingredients or excipients, and different manufacturing processes. And there are so many places where things could go wrong. So usually in between lab and commercial scale production, there's pilot scale. It starts in the lab by making slightly larger batches, while also characterizing the processes that will be eventually employed to mass manufacture. Production volumes at pilot scale are closer to commercial scales, including final formulations and packaging. Taking a drug through pilot scale before getting to commercial scale mitigates risk and avoids unnecessary setbacks.
Unlike small molecule drugs that are completely synthesized chemically in the lab, large molecules, usually proteins, for biologic drugs, are synthesized and grown inside living cells. So biologics manufacturing involves as much biology as manufacturing. It involves upstream processing, where a cell is forced to synthesize the desired protein, and explosively growing the number of cells with the protein, and downstream processing, where this large number of cells is separated from the proteins, which is then formulated into the final drug. Zooming in, upstream processing includes expressing the proteins in a host cell, preparing it to bond with the drug target in the body through glycosylation, and culturing or multiplying the cells in a bioreactor. Downstream processing involves centrifugation to separate the proteins from the host cells, chromatographic and other filtrations to filter out virus vectors and other impurities, and finally, mixing the protein with inactive excipients in the right conditions to formulate the final drug. In this video, we'll be taking a look at protein expression.
Protein expression actually starts with recombinant DNA, a strip of genetic material synthesized artificially. It's usually DNA fragments from different species spliced together. In the context of biological drugs, recombinant DNA is used for one thing: to carry instructions into cells to produce or express a specific type of therapeutic protein. Recombinant DNA thus leads to recombinant proteins. But the DNA requires some help to pierce a cell wall and enter it. Enter a vector, usually a virus mounted with the recombinant DNA. By its very nature, the virus is adept at piercing the cell wall and invading cells, getting the DNA into it. Once inside the cell, protein expression occurs in two phases. First, the information in the recombinant DNA is copied onto another type of genetic material, a messenger RNA or mRNA, through a process called transcription. mRNA then prods the cells to express the target protein through a process called translation. So protein expression is transcription plus translation. The therapeutic protein or biologic thus expressed is not yet ready for prime time. It needs some post-translational modifications to stabilize it and prepare it to bond with the drug target inside the body, something we'll look at in the next video.
Unlike small molecule drugs that are completely synthesized chemically in the lab, large molecules, usually proteins, for biologic drugs are synthesized and grown inside living cells. So biologics manufacturing involves as much biology as manufacturing. It involves upstream processing, where a cell is forced to synthesize the desired protein and explosively growing the number of cells with the protein, and downstream processing, where this large number of cells is separated from the proteins, which is then formulated into the final drug. Zooming in, upstream processing includes expressing the proteins in a host cell, preparing it to bond with the drug target in the body through glycosylation, and culturing or multiplying the cells in a bioreactor. Downstream processing involves centrifugation to separate the proteins from the host cells, chromatographic and other filtrations to filter out virus vectors and other impurities, and finally, mixing the protein with inactive excipients in the right conditions to formulate the final drug.
In this video, we'll be taking a look at post-translational modifications. A protein freshly expressed in a cell through mRNA translation is very unstable, susceptible to even small changes in temperature, pressure, or pH. As such, they don't last very long in test tubes during in vitro studies. Once inside the body, they'd show poor bioavailability, which means they disintegrate before they get to the target site at the cellular level. Trying to solve this instability problem starts by looking at the protein structure, which is actually a long string of amino acids, each depicted by an English alphabet. A popular post-translational modification to stabilize a freshly expressed protein is to attach carbohydrate chains or glycans to some of these amino acid sites, a process known as glycosylation. N and O glycosylation, where the chains are attached to the N and O amino acid sites, are the most common. Nobody is quite sure how glycosylation works, but it makes the resulting protein rugged enough for the rigors of in vitro experiments. Not just that, once inside the body, glycosylation also plays a crucial role in cell signaling, telling the cells that it's okay to take in a foreign object, in this case a therapeutic protein, without attacking it with the defenses of the immune system, vastly improving the biologics' bioavailability. Glycosylation, a crucial post-translational modification which stabilizes a newly expressed protein.
Unlike small molecule drugs that are completely synthesized chemically in the lab, large molecules, usually proteins, for biologic drugs are synthesized and grown inside living cells. So biologics manufacturing involves as much biology as manufacturing. It involves upstream processing, where a cell is forced to synthesize the desired protein and explosively growing the number of cells with the protein, and downstream processing, where this large number of cells is separated from the proteins, which is then formulated into the final drug. Zooming in, upstream processing includes expressing the proteins in a host cell, preparing it to bond with the drug target in the body through glycosylation, and culturing or multiplying the cells in a bioreactor. Downstream processing involves centrifugation to separate the proteins from the host cells, chromatographic and other filtrations to filter out virus vectors and other impurities, and finally, mixing the protein with inactive excipients in the right conditions to formulate the final drug.
In this video, we are looking at the last step in upstream processing, cell culturing. The process of taking a cell containing a glycosylated protein and explosively multiplying it to mass produce the protein. At the core of cell culturing is this piece of equipment, a bioreactor, used in all kinds of biological processes, from sewage treatment to synthesizing novel protein therapeutics. At its center is a big hollow drum called the reactor tank, in which the cells with the expressed proteins are introduced. The tank also contains a nutrient-rich medium or syrup which the cells feast on as they multiply. There's usually a way to aerate the medium because oxygen is crucial for cellular metabolism, and a paddle or agitation system to stir up the cell and the medium mix. There's insulation around the bioreactor to maintain temperature, and a way to let out the used medium or effluent. The aim of the bioreactor is to improve titer, which is the concentration of target protein in grams per liter of the solution within the bioreactor. Most commercial biologic setups gives a titer value between 2 and 4 grams of protein per liter of solution. Most biopharma companies around the world are in the pursuit of increasing this value, or titer improvement, and they employ a host of strategies, like using more advanced techniques to express more rugged proteins, using better engineered growth media, or by using process analytics techniques to improve manufacturing parameters like temperature and pressure. Because an improved titer means lower costs and getting your biologic drug to many more patients fast.
Unlike small molecule drugs that are completely synthesized chemically in the lab, large molecules, usually proteins, for biologic drugs are synthesized and grown inside living cells. So biologics manufacturing involves as much biology as manufacturing. It involves upstream processing, where a cell is forced to synthesize the desired protein and explosively growing the number of cells with the protein, and downstream processing, where this large number of cells is separated from the proteins, which is then formulated into the final drug. Zooming in, upstream processing includes expressing the proteins in a host cell, preparing it to bond with the drug target in the body through glycosylation, and culturing or multiplying the cells in a bioreactor.
The harvest obtained from a bioreactor at the end of upstream processing has a lot of the therapeutic biomolecule which will form the core of a biologic drug. There's only one problem. It has a lot of other unwanted things too, like host cell fragments, virus vectors used to penetrate the host cell, and growth media. So there's a lot of downstream processing to be done before the bioreactor harvest becomes a fully formulated biologic drug. Downstream processing starts with primary capture, the process of separating the desired biomolecules from bulk or heavy impurities, like host cell fragments. A popular way to do this type of high-level separation is centrifugation. When you rotate a mixture at high speed inside a centrifuge, The heavier components tend to move towards the edges and the bottom of the centrifuge, whereas the lighter components tend to float at the center. Next up is purification. Basically, several rounds of filtration to separate the biomolecule from other smaller impurities. In biological setups, filtration is done using many different types of chromatography. At its core, chromatography involves running a mixture through a system of filters, or plates or columns, whose physical or biochemical properties aids in filtration. For example, in size exclusion chromatography, the mixture is run through a column containing a particular gel. The physical properties of the gel at a molecular level allows it to trap small particles, whereas the larger impurities bypass the column and go on. There are other types of chromatography which relies on the biological affinity between molecules. A popular example of this type of affinity chromatography is protein A chromatography, used in the filtration of monoclonal antibodies or MABs used in many biologic drugs. MABs are known to have a high affinity for protein A, a type of protein usually isolated from bacteria. So if a mixture containing MABs and other impurities is run through a protein A column, the MABs will get stuck, while the impurities won't. Once the filtration process is complete, MABs can be detached from the protein A column by washing it in an acidic buffer solution. Once the therapeutic biomolecule is isolated and available in large enough quantities, begins the process of formulating it into a finished biologic drug, where a drug substance transforms into a drug product. Formulating a biologic drug isn't just adding some taste and color to the drug substance. Biomolecules like proteins are notoriously unstable and by itself will not survive the rigors of storage and transportation. So formulation involves trying out hundreds of binding agents and stabilizers to improve the drug's shelf life. And most protein-based biologic drugs cannot be taken orally because the stomach acids would destroy the protein. Instead, they have to be formulated as injectables, given intravenously or into the muscles, or as aerosolized nasal sprays. Through harvest, chromatographic filtration, and formulation, downstream processing converts a biomolecule into a finished biologic drug. Maintaining quality across a pharmaceutical organization is the responsibility of two related yet distinct teams. Quality control, which deals with product quality, and quality assurance, which deals with the quality of processes that are in place across the organization. They can also be thought of this way. Quality control is responsible for detecting errors, while quality assurance is responsible to make sure that errors don't occur in the first place. So the quality control team is responsible for collecting samples from the production batch and checking it to make sure the active ingredients are in the right concentration, impurities are within limits, and that the product will dissolve in the human body as expected. In other words, the quality control team is responsible for making sure that the product meets approved specifications. Because of the extensive experimental nature of the work, quality control is usually performed by personnel with technical backgrounds, like in molecular biology or biochemistry. A quality assurance person, on the other hand, is a stickler for processes, always emphasizing the importance of documenting the way things should be done and doing things exactly as documented. For a quality assurance person, the emphasis is always on behavior which affects quality. Call it the quality culture of an organization, and no detail is too small. And in a pharma setup, QA is responsible for maintaining quality culture through production processes, by making sure only trained personnel and qualified equipment are involved, at the warehouse including receiving raw materials, for maintaining company-wide records and documenting standard operating procedures or SOP, and investigating what happened in the event of customer complaints, recall, or out of specification events. In fact, no production batch can be released until the QA department signs off on it, thereby taking responsibility for the batch. Given this responsibility, QA personnel frequently conduct internal audits of the company's processes and get people and equipment ready to meet regulatory inspections. Let's say you're in the mood for some pizza. You open a food delivery app, go to your favorite restaurant, and pick the pizza with all your favorite toppings. And just like that, a whole process kicks into action until the pizza is delivered hot and fresh right at your doorstep. You open the box and you find out it's not your favorite pizza at all. The toppings are all wrong, and even the size is not what you asked for. In scientific parlance, this is what's called an out of specification or OOS event. But who exactly is responsible for this OOS event? Was it the app which showed you an option that just wasn't available? Maybe the restaurant which didn't have what you asked for and instead substituted with something they had. Or maybe it was the delivery boy who gave you someone else's order. You need an OOS investigation to find out. If OOS can happen with pizza delivery, it can surely happen with much larger and more complicated processes, like drug development and production. Sometimes the API, in-process samples, or even the final product are found to be out of specification when tested. And just as before, the errors causing OOS in the drug industry can lie in many different places. From the quality control analyst who did the testing, to the production team that manufactured it, or even the person who sampled and handed over the samples to QC. To maintain consistent quality of the final drug product, it's crucial that the root cause of OOS be investigated and proper corrective and preventive action, or CAPA, be put in place to avoid reoccurrence. As important is to follow quality by design principles in drug development and manufacturing processes, so they yield fewer OOS, and even if they occur, the root cause can be easily identified. For data to be reliable, it must follow all principles of data integrity. Alcoa, upgraded to Alcoa Plus, forms the gold standard for compliant data and records. A in Alcoa stands for attributable. Who recorded or modified the data, and when? Are you wondering what's wrong with this? Data has been modified, but it's not attributable. No one knows who made this change and when. Here's the correct way to make the same modification. L in Alcoa stands for legible. Data must be neatly written and easily readable by everyone. This is an example of data that's not legible. Data must remain legible for the entire life of the document. You should never need to reach out to the author of the document. Now let's look at what's wrong with this picture. Change is attributable here, but the original recorded data is not legible. Here's the correct way to make the same modification. Data should also be permanent, so erasable ink pens and pencils should not be used. C in Alcoa stands for contemporaneous. Data must always be recorded in real time. When your activity or test is completed, record the data immediately on an approved document. For most electronic documents, all data is date and time stamped automatically. O stands for original. It is the first capture of information, whether on paper or electronically. For this reason, data should not be recorded on a scrap piece of paper, a rough notebook, or a post-it. Always record on an approved document. As an example, a raw chromatogram stored on a computer is original, but a printout of the same chromatogram cannot be considered original as it may have been changed before being printed. However, a printed output obtained from a device that does not store data, such as a pH meter or a weighing balance, can be considered original. The last A in Alcoa is for accurate. Data must be correct, truthful, complete and reliable. It must be without errors. All corrections made should also be well documented. In addition to Alcoa, data must also be complete, consistent, enduring, and available. These 4 additional parameters makes the plus in Alcoa Plus. For data to be complete, all results, including failed results, should be recorded and archived. For data to be consistent, it should be recorded in the expected sequence of execution. The date and time should be evident during an audit. Data must be enduring. That means it should last throughout the data lifecycle. Corrupted electronic data or faded paper records are examples of non-enduring data. And finally, for Alcoa Plus, data must be available for review and for inspection. Data must be stored such that it can be retrieved from an archive or restored from an electronic backup quickly. Alcoa Plus, an easy way to remember how to maintain data integrity.